All right, David, I think we're going to get started. Okay. Good evening, everyone, and welcome to the February Neighborhood Health Series. We're happy to see such a large number of you here with us since we pivoted to a virtual format with the onset of COVID-19. So we hope you're all very healthy and well. And we also hope that we will get back to doing this series in person very soon. I'd like to just take you through a couple of quick facts, some Roseman trivia before we go any further. For those of you that might not know much about Roseman, Roseman is the largest not-for-profit private university in Nevada. It started in Henderson in 1999, so we just celebrated our 20th anniversary. We have over 550 employees across 3 campuses, Utah, Summerlin, and Henderson. And we have over 1,500 students currently enrolled. We are built on an innovative educational model, which is the Roseman 6-point mastery learning model. And David will, as a faculty member, probably demonstrate some of those facets tonight as he takes you through this presentation. We conduct educational, clinical, social, and bench research in a variety of critical healthcare topics. And 83% of our students receive some form of financial aid. We have one of the best and lowest default rates across the country for our students, less than 1.5%, which means that our students borrow to get through their programs and they quickly repay their loans because they are employed very quickly after graduation. We have a diverse student body drawing from over 43 states and 16 countries worldwide. And as you probably know, we have a College of Dental Medicine, Nursing, and Pharmacy. And right now we're in the process of building our College of Medicine and College of Graduate Studies. So with that, I'd like to thank you all again for being here, and thank you so much to our sponsor, Clark County Credit Union. We could not do this series without them. They are an incredible community partner. We're very grateful. And thank you very much to my team. Before I introduce our speaker, just a couple of housekeeping things. We most likely will take our questions at the end. However, those of you that have need clarification on some of the subject matter, feel free to enter your question in the chat feature at the bottom here. You can see it if you mouse over on the bottom of your screen, you'll see the chat and you can enter your question there. We are recording this event tonight, and this will be posted on the speakers.roseman.edu site as we do all of them. And so you can always go back and find those and share them with those that you'd like. So with that, I'd like to introduce our speaker. Dr. David Rawlins serves as Interim Assistant Dean for Research in Roseman University's College of Pharmacy. He received his PhD in organic chemistry from Stanford University following earning an MS in chemistry and a BS in microbiology from Brigham Young University. After graduation, he joined Bristol Myers Squibb as a medicinal chemist, working on drugs to treat oncology and immunology diseases. After 8 years at BMS, he joined Lexicon Pharmaceuticals, researching drugs to treat diseases in oncology, metabolic disease, and ophthalmology. Before joining Roseman University of Health Sciences, he was executive director at medicinal chemistry and led both chemistry research teams as well as the Chem Informatics group. His interests range from traditional medicinal chemistry to building software tools to aid research. His work has resulted in 18 journal articles, 17 patents, and 3 drugs that have entered human clinical trials, one of which has been approved in the EU. Please join me in welcoming Dr. David Rawlins.
Thank you so much, Vanessa. And honestly, we couldn't have this series without Vanessa either. She is wonderful in organizing this and getting volunteers to help out. It's wonderful to see all of you. Well, I shouldn't say see all of you. I see your screens. I recognize names from current students to past students to administration and our community friends as well. So it's great to have you here. I can see you on the screen if you want to have your videos open. That will help me feel like I'm talking to someone, but I understand that not everybody likes to do that. The other thing I'll point out just at the very beginning here is if you look carefully, there's 2 of me. One of them is called slides, and that's the one that will have that is sharing the slides. If you want to pin that one to your screen, that one will be there. So you can always have the slides regardless of who's speaking if it jumps around. Um, with that, I also want to thank Clark County Credit Union for their support. Without that, this wouldn't be doing what it is. And I want to turn the time over to Craig Farley, who's here from Clark County Credit Union, to say a few words.
Thank you, Doctor. As the medical field credit union, Clark County Credit Union, we are very proud to help sponsor Roseman's Neighborhood Health Series. If you should ever need banking services, we've been around since 1951. We're one of Southern Nevada's oldest businesses. We are better than a bank because we're owned by our members, not for profit, and we give our profits back at the end of the year. Since 2001, our 47,000 members have shared in a little over $68 million back in their accounts. You can find us at ccculv.org, and you can even open your account on our website. I hope you have a wonderful evening tonight. Enjoy the presentation. I'm turning it back over to the doctor. Thank you.
Thank you, and clearly, thank you for your support. One other thing I will mention, I do have the chat open here. I feel like I'm teaching again. I've got all the screens and the chat and see all the things. I'll be watching it. I may not answer everything right off. I may push some to later on, but if there's something that's really important for clarification, I will actually be able to see that and be able to answer that, hopefully in as close to real time as possible. So tonight, I've probably bit off a bit more than maybe we can chew, but we'll see how it goes. There's a lot of material in these topics that we're going to go over. So we will be talking about them at, I'm trying to give you a flavor of what's going on, maybe a little bit. We're not going to dig too deep in many of these. Each of them could be its own event, honestly, because there's so much material to talk about. But what I will do is, since I don't know where all of you are coming from, I will review just very basics of genetics, just enough that for what we need to know for what we're going to discuss tonight. We will then talk about the genetic markers, what those are, what they are, and how those are used. This is where we'll talk about forensic investigations and ancestry, that kind of thing. And then we will talk about how they're used for understanding how our body responds to medications. And I'll give you some examples of that. So again, please feel free to send anything in the chat. We'll try to have this be as interactive as possible. All right, so what defines us? Many of you you all know this, I am sure. DNA stands for deoxyribonucleic acid. Now, I'm not going to make you draw the structure of that. I'm not sure I would want to draw the structure of all of that, but each of the cells in our body has DNA in it, and it's like the recipe book. It has all of the recipes for everything that needs to be made in our cells. We have to maintain that recipe book, keep it, its integrity, so that we can maintain and grow our cells as we go along. So we have ways of replicating that. We have ways of repairing it if there's problems with it. And all of that is critical in order to maintain our cellular functions. From our DNA, we take out recipe cards, which are little copies of the DNA. That's our RNA, ribonucleic acid. The ribonucleic acid is what contains the instructions for one thing. Now, some of you may recognize this a little bit. If I were to put a little M in front of that, you might have seen that when in a discussion of COVID vaccines, such as Pfizer or the Moderna vaccine. The mRNA is just simply a ribonucleic acid, which contains the code for one of the proteins that's on the surface of the virus. So that's similar to what would happen in our body when we want to make a protein. We're going to make some copy of the mRNA and have it go through that. And then the proteins are synthesized and have some kind of cellular function associated with them. So if we were to look inside of a cell, we each have 23 pairs of chromosomes. They sort of look like this picture. This is obviously not a microscopic view of it, but a representation of it. Those chromosomes are contained in the nucleus of the cell, and they are tightly wound pieces of what we call chromatin. Chromatin is a long string of DNA that's wrapped around a nucleosome, or histones, if you've ever heard of those. All of that's done to try to pack it in order to protect it from breakage, to protect it from being expressed if we don't want to make the proteins. And at the time of cell division, we have to pack it that way as well in order to be able to separate it out into the daughter cells. Now, in the DNA, we have genes, and it's estimated that we have about 20,000 to 25,000 genes in our DNA. That gene, as we'll talk about on the next slide, is the code for one functional unit. Those genes are made out of nucleotides. Nucleotides are the individual pieces that are stitched together to make the DNA. You may have seen things like this where you talk about C, A, T, and G. That's the abbreviation for some of those nucleotides. So the nucleotide is the smallest unit, makes up the DNA, which has the genes and goes on into the chromosomes. So as I said, the gene is the segment of the DNA which is going to code for some functional component. It's usually a protein, not always, but that has the instructions of how that protein should be stitched together to try to make it have its function. So we often call this the unit of heredity, where that is how we pass these different traits on is through the genes. In most of our cells, we have 2 copies of every gene. 1 copy from mom, 1 copy from dad, with the exception of those that are found on the X and Y chromosomes. There are not exactly the same ones between those, and that makes up a difference, which some people say is Venus and Mars. So we have these extra copies. We try to shut off some of those at some time. There's a lot of regulation that goes into it. We're not going to go into that tonight. But each one of the copies of a gene is called an allele. So you may have heard that term before, but an allele simply means 1 copy of the gene. Now, when we have the reproduction occurs, when mom and dad are making a child, we have 23 chromosomes in the egg from mom and 23 chromosomes packed inside the sperm head from dad. When those combine, then we have 46 chromosomes, not pairs, but 46 chromosomes. And those carry the traits that we see and are often evident. Now I've given you a picture of a mother and a father and a daughter here. I think you can probably see some of the traits that are passed from mother to the daughter as well. This happens to be my granddaughter, so that's why it's there. But if in case that isn't clear enough for you, I do have one more picture. If you look at this, this is a daughter, a mother, and a mother-in-law of mine. So these 3, if you look at that, there's some pretty obvious traits there that carry through that are coded for or expressed by those genes. Now, at the time of cellular division, which is required prior to the reproduction to form a new organism, the DNA must be replicated. Now, the DNA contains two strands that are wound around each other. I'm sure you've seen pictures of it before. Those strands are pulled apart and they serve as a template for new daughter strands. In that process, there's nucleotide bases that are brought in and stitched together. They match, they pair with specific ones, and when they pair with that, that allows you to keep making a DNA molecule. Interestingly enough, the enzyme which stitches these together does so putting about 1,000 nucleotides a second together in that DNA molecule. That's pretty fast as far as I'm concerned. However, there are times when it can make mistakes. We have mechanisms to try to repair those mistakes, but there's also other mechanisms which can bring damage into the DNA. Some of these you've heard about. Laying out in the sun, UV light can damage the DNA. The replication process itself can introduce errors or problems. X-rays, why do we have to wear a lead blanket trying to protect the cells that could be damaged by the X-rays? And oxygen radicals can do that as well. And I've given you, over here on the side, there's just some information there about some of the different things that can happen with the damage. Now, as I said, we have mechanisms, very sophisticated mechanisms and pretty impressive to actually go through and repair all of the DNA errors as quickly as it can. If those errors are not repaired prior to DNA replication, however, the errors then get incorporated into the DNA. Because once you've replicated, it matched it as it put it together, and it can't tell what it was supposed to have been previously. It's too late at that point. When those permanent changes are made, we call those mutations. Now, a lot of times people think of mutations and they think of zombies and, you know, all of these bad things that happen or whatever. The reality is that most mutations are not harmful and don't really do anything. We have a lot of segments of DNA which really don't code for anything. They're just there. And if there's a change, a mutation in those, that doesn't really, we don't see it. It's not expressed. Sometimes changes are done in the code for the protein without making a change in the amino acids. And again, we don't see that, an effect, at least in the body. So a lot of times mutations are not a big deal. Although the ones that we hear about the most are the ones that are a big deal. So let's talk just a little bit about how this genetic inheritance occurs. So I've given you a simple drawing here of a mother and a father. The father has one copy of a bad gene. That's why it's hashed there. So he has one copy that's good on the right-hand side here, and one copy that's bad, depicted here with the little lightning bolt that's going down into the chromosome. Mom has two copies that are good. When, through sexual reproduction, there are two possibilities when these chromosomes mix. There's one chromosome from dad can go to this side or the other side, and one from mom can go there as well. One child then becomes a carrier and has one copy of the bad one, and the other child has no copies of the bad one. It just is not carried through in that side of the line. Now, this is very, very simplistic, and it gets much more complicated pretty quickly. I'm just showing you here now what happens if mom carries it too. So in this case, each of them has one copy of a bad chromosome. And so those can partition out as they're separated out. And one child will get two copies of the bad chromosome. And so they will have no good copy of it. Two of the four are going to end up carrying one bad copy of it, and the fourth one ends up with no bad copy. That's statistically speaking. Now, it's obviously possible that the inheritance doesn't quite follow statistics, and you may have more than just 1 out of 4 children that carry or that end up with the gene. So as I said, it gets a lot more complicated because most of the traits and things like that that we observe are influenced by multiple areas in the DNA. And so it can be really, really complicated. Plus, if it's a gene that's on either the X or Y chromosome, it's a little bit different as well, but I'm not going to go into and chart that out for us here. Now, some of these variations or mutations have been around long enough that they're present in a larger percentage of the population than we would think that they ought to be. So the rule of thumb is if it's just a random event, it's usually present less than 1% in the population. But if we have a mutation that's present in greater than 1% in the general population, we call that a polymorphism. Polymorphisms are all throughout our DNA. And another way of thinking of these is genetic markers, okay? Something that will allow us to detect or tell different strands of DNA apart. There are a couple different types of polymorphisms. One set is called the single nucleotide variants, often called single nucleotide variants or single nucleotide polymorphism. Sometimes you may have heard of SNPs, but currently the terminology is moving to single nucleotide variants. Now, that's a change in just one single nucleotide, and that could be in an area for the protein code, or it could be in some other area where it doesn't code for the protein. It could be anywhere in that DNA. But as long as it's present there and we can detect it as different from everybody else and greater than 1%, we say, hey, this is something that's been around for a long time and we can look at it. There are also structural variants. Structural variants are insertions or deletions or even extra gene copies. And so those are a little bit different. So we can have just the change in one base or larger situations. These polymorphisms can serve as a fingerprint, if you will, a DNA fingerprint. If you look at enough of them, you can see how something is related to something else, because if I have all of the same polymorphisms as someone else, we're pretty close in relation, because there hasn't been time to introduce a whole lot of other variants. And I'm showing you a picture over here. This is just one way of depicting it. We call this a heat graph. And what you can see, I hope you can see anyway, is that these over here on the left side have a lot more red in them than the ones on the right side. And you can see that the ones that are more blue are more related to each other than the ones that are red, and so forth. And so as we're looking at all of these, that's really what we're kind of doing, is we're trying to look at a whole great big set of these little teeny changes in the DNA to try to have it be a fingerprint and tell us exactly what's going on. Now, what I've shown you so far, we've been talking about the passing of genetic material from mom and dad to child, and then from mom and dad on down, and it just keeps going down to the individual that we're talking about. That genetic material is all associated in the nucleus. So what I've been talking about is the nuclear DNA. Well, interestingly enough, the mitochondria, which are kind of the powerhouse or the power plant in our cells, they also contain DNA. Now, it's much less DNA than is in the nucleus, but they do have DNA. It creates an interesting phenomena because the only source of your mitochondria was your mom, because the mitochondria is present in the eggs, but not in the sperm. So every mitochondria that you have contains the same DNA or very closely related DNA to what your mom had. So we can trace a maternal line by looking at the mitochondrial DNA and following it back from one mom to the other. Notice it doesn't jump and go to any of the other moms because it's just going from the mom to the mom to the mom, just straight back. Likewise, we can do this with the Y chromosome as well. And this would be our paternal line. The Y chromosome is only inherited from the dad, and we can follow that on back through that. You may have heard of something called Mitochondrial Eve. The principle behind Mitochondrial Eve is that as we sequence the mitochondrial DNA, we can trace it back from one woman to another woman to another woman and just keep going back. It all comes back down to this area in sub-Saharan Africa, which is referred to as the L0. That's the origin of that mitochondrial Eve. Now, you can have your mitochondrial DNA sequenced and see which of these different groups it fits into based on what changes have been passed down from one mother to another mother to another mother. So my maternal line is what is known as J1C1. It's a branch of J, which is present here in Europe, and that goes back to a woman who lived there nearly 10,000 years ago in Europe. And then she came from N, which came from L3, which came from L1. So you can trace it back that way. Just looking at the collection of all of these little individual changes in the mitochondrial DNA and matching how close they are to each other. Likewise, we all have a little bit of Neanderthal in us. So the Neanderthal DNA was incorporated in through crossbreeding between the Neanderthals and the hominids. So we have some of that in us. And now the amount of Neanderthal DNA that we have varies in each person. Now, there are some variants which are present in over like half of all of us. And there's other ones that are very rare, less than 2%, and specifically in those of Eurasian descent. Interestingly, this section of DNA, the Neanderthal DNA, is rarely found in the code for the proteins, but rather in regulatory areas, so controlling how much something is expressed. The other thing that I found very interesting about the Neanderthal DNA is it's very often associated with our immune systems. In particular, it's associated with how we interact with viruses. That will come back in just a minute. We will talk more about that. Whoops. Now, I've given you a basic overview of genetics. Many of you, I'm sure, have heard of the direct consumer genetic testing that's available. There's all kinds of different companies that will do this. Most of them are centered around ancestry and being able to trace back your heritage, your relatives. There are some others which are specializing in some other things. This is relatively cheap these days in comparison to what it used to be. And a lot of people have it. If you look at how much over here, this is millions of people here have their DNA that's already sequenced, and these are 3 different companies. So if you can't read it, the green is Ancestry DNA, the pink is 23andMe, and then everybody else is lumped together in the gray. So millions and millions of people have done this and have genetic information with them. Now, one challenge with this genetic information is the results may not be as clear-cut as many people think that they are. So you may have something that suggests that you're going to have some kind of an effect, but that whether that is truly expressed, whether you're really going to have that, it's just suggestive. It's something that was associated with it in some frequency, but it is not an all or nothing. So for instance, let me give you an example. My genetic tests say that I have a very high likelihood of having ovarian cancer. I have not lost any sleep on that. I have not even worried about it. But because of my genetic marker, which is associated with ovarian cancer, the test comes out and says, you have an increased likelihood of ovarian cancer. So that's why I say you have to be kind of careful with it and take it and look at it a little bit more carefully. When they first started doing these tests, there were a lot of reports that were given without the context of how reliable it is or anything like that. And because of that, the FDA cracked down on it a lot. And so primarily what you can get now is ancestry things and a few health reports that the FDA has allowed. Now, one exception is just a few years ago, and 23andMe was granted authorization from the FDA to actually offer what they call a pharmacogenetics report as part of their services. This has different genes that are associated with drugs that they can give you a report on and tell you more about it. There's only about 6 of them right now that the FDA has authorized them to talk about. Now, for their purposes, they have done this such that in order to have that kind of report, you have to have a yearly subscription with them. And if they add more reports, they would give it to you. Interestingly enough, and if we have time, I'll show you at the end, if you even do the simplest one on 23andMe, they actually test and sequence all of it exactly the same way as the other assays. And if you know how to look at your raw data, you can sometimes get all of the same information. Now, I have not personally done this newer service, and so I can't tell you for sure that they haven't removed some of the genes from the regular panel that they screen. It's possible that they've done that, but for most intents and purposes, you can actually get a fair bit of information beyond what they tell you if you know how to look for it. All right, so that's a broad overview of kind of the genetic markers, what they are, these little changes in the DNA, and how we can look at those and make associations from one thing to another based on how similar that DNA is. And so I want to give you some examples of the utility of these. Now, there's really 4 different things that we're going to talk about tonight. The first one is identifying a person, like forensic evidence, like from remains or some other evidence that was left at a crime scene. Identifying your relatives, identifying susceptibility to disease, and then also how you might respond to medication. So just as an example, for instance, this dapper looking man from England was a not so smart robber because he went into somebody's home and ate half of a peach and left that half there. And they were able to come in and find the peach where he had only eaten half of it, isolate his DNA from it, and catch him. So that's the forensic evidence part of it. So let me tell you a little bit about how that works. So this is showing you the different chromosomes. And from what I read, and I'm not an expert on this, but from what I understand, there's 13 different sites that the FBI can use to try to match one set of DNA to another set of DNA. And you see that they're spread throughout the different chromosomes. There are all these yellow areas. And they're looking at in that particular area to try to see how closely there's a match. And we're going to do an example with this. So in this example, there was a hair found in a glove. And that hair had enough of the root of the hair in it that they could amplify it up and sequence the DNA. And they came up with these two sequences, which were in that hair. Now, I should point out, this is only looking at this one site in the chromosomes, this D21S11 site. Now, there's 3 suspects, and they get some DNA, either hair or a cup that they drank from, whatever it is, and they have done that and looked to try to see which of these have the same kind of DNA in them. So take a look at that and think about it a minute. Which of these suspects, 1, 2, or 3, do you think could have left the hair in the glove? You can type in the chat. If you want to unmute, you can yell it out too, however you want to do it. I just want to give you time to think about it. Okay, I've got a couple that have said 2. Why not 1? Oh, there was somebody that said 1 or 2.
It could be all of them equally.
Well, with the exception of 3, Omed, because 3 does not have what is this green area that I've highlighted. So
Yeah, sorry, that's what I meant. I meant it could be any 1, 2, or 3. Oh, actually, no, you're right. Yeah, I mean it could be 1 or 2 equally. Sorry.
Right. Well, it could be 1 or 2. 3 doesn't have that one section. Now that doesn't mean it's not 3. That's just because we're only looking at one site, right? So maybe we just missed it. Maybe we didn't sequence it. So let's look at another site. So here's the TH01 site. Now the sequences, I've highlighted these in green to try to help you see it a little bit faster than, because you don't have it on paper in front of you. Here's the evidence from the glove. Which of the suspects would match this one? I see somebody saying peace. Yep, a number of people saying
2.
Yep, number 2. All right, that's the only one that has the blue area in it. All right, let's do a third one. How about this one?
Two.
Yep, just 2. Yep, there's not, you don't see those sequences in 1 or 3. And then the last sequence here, we're only going to do 4 of them. Hopefully by now you're catching on. It could be either 1 or 2. All right. So we looked at 4 sites. 2 of them said it was 1 or 2, but 2 of them said it was only 2. Which suspect do you think is the most likely to be the suspect in this?
Suspect 1.
Okay, so suspect 2 matched in all 4, right? So suspect 1 matched a little bit, but suspect 2 matched in all 4 of them. And so if we look at that by statistics, there's actually a 1 in 10,000 chance that that was just by random. So essentially, I'm pretty sure that that is 2 for subject, the one that did it. Now, if we were to go on and look at all 13 of those, and all 13 of them matched subject 2, that's actually a 1 in 10 trillion chance of being just random. So sometimes, and you may see these on the crime shows on TV or whatever, what's the likelihood? Well, it's a 1 in 9 billion or something like that. That's basically what they're looking at here, is how frequently did that suspect match and go with whatever sample we had. If we have more than one sample and they're matching all of them, that even increases the confidence that we have that it is who we are fingering. All right, so that's an example of forensic evidence. Now let's talk a little bit about ancestral tracing. So this is the same idea where you're looking at all kinds of different sequences in the chromosomes and trying to match how close you are to someone else. So this is from 23andMe, and this happens to be my DNA. Now, what you see here is it's color-coded, and I know it's a little small, it's a little hard to see, but the different blues are either from the British and Irish heritage, French and German, Scandinavian, or other Northwestern European ancestry. So you see they have that all mapped out there. There were a lot of different heritages that I did not have, and other colors that don't show up. Basically, they said I'm 100% European. Now, if you add up everything here, I figured out that I have, from this analysis, 57% of me is from the United Kingdom, 32% from Germany, and 12% from Norway. Okay? Well, I did this with Ancestry DNA as well, and I get a little different result. So in Ancestry DNA, they're looking at some different markers, so it's a little hard to totally add everything up exactly the same way. But according to them, I'm 72% from the United Kingdom, 4% from Germany, and 18% from Norway. Now, they then look at everybody else who's had their DNA sequenced and tell you how much you have in common with them and try to then say, this is a cousin. And I'll show you that here in just a little bit. Now, I happen to have a fair bit of my own family history done, and I'm showing you here a fan chart from my family history. Now, if you're not used to seeing a fan chart, what this is this is me, this is my father, and this is my mother. This is my grandfather on my father's side, my grandmother, grandfather here, and grandmother, and so forth as it goes out in this fan. Now, what I've done is I've color-coded them based on where they were born. This is as good as I could do to try to determine ancestry in that sense. Now, obviously, that's not going to be as precise as DNA. I get that. But that's as good as I could do, and just did a rough estimate of that. From that, I come up with 69% from the UK, 22% Germany, and 13% Norway. Not too far off. The Germany one, I don't know why Ancestry has me so low in Germany. Maybe I didn't have enough chocolate that day or something. I'm not quite sure. But generally fairly close. One of the things that I was really kind of interested in, though, in one of these more recent analyses, which I'm showing you here, is this right here. In my German heritage, they pinned it to an area in Germany called Schleswig-Holstein. And I don't speak German, so I probably slaughtered that. Now, if you were to trace my ancestry and what I know here through the Rawlins line that goes back from my dad and the Ovard line from my mom, and you were to trace that all the way back, you're not going to find Schleswig-Holstein anywhere in there. But they say I have Ancestry Farm there. Well, let me explain. This is my grandfather on my dad's side. My grandfather was born, as the son of John Henry Hansen and Caroline Jensen Weeding. Now, Caroline Jensen Weeding has Norwegian ancestry. John Henry Hansen has German ancestry. So my grandfather was born a Hansen. At the age of 10, he was adopted into the Rawlins family. So I actually have no Rawlins DNA in me. What I do have is from the Hansen line. Interestingly enough, if you look at this, John Henry Hansen, his mother Maria, and his father Klaus were all born in Schleswig-Holstein, Germany. Now, unfortunately, that's as far back as I've been able to get that particular line, my biological line, if you will. It ends right there, and I haven't been able to trace it back any further. I'm still hoping and keep looking to try to see if there's any hints that come through that would help me do that. But my point here is to show you that that sequencing has gotten specific enough that they can pinpoint it to certain areas in these countries even as to where your ancestry is from. All right, now I said we would talk a little bit about how they do this, and they're doing that based on how similar your DNA is to someone else. Now, they use something which my wife asked me what this meant this morning, and I couldn't answer, but I can now. They use units that are called cm, which are centimorgans. All it's doing is saying this is how close the DNA is. So the more centimorgans you have, the closer it is to someone else. Now I'm showing you this chart. So if this were me, I should match 100% of my DNA. Yeah, you can roll your eyes. My daughters would roll their eyes. That's kind of a dad joke, but all right. If you look at it then, my child would have 50%, my parents would have 50% match, and then it would go on and it goes back through and can predict other things, but you have to be a little bit cautious. If you look carefully, for instance, if you look at this percent DNA, that's a first cousin once removed at a 6.25, and here's a great grand uncle or aunt, which is also 6.25%. So just by percentage, it could be either one of those, but you can tell how close it is to something else by the markers, how some markers are inherited more recently than others. That kind of thing can help you out in trying to pinpoint more precisely. Now, there was a question about the commercial tests versus nationality versus ethnic and racial background. That ethnic and racial background from the DNA ties back to a particular area where that was. Now, in some cases, they can't do that. So if I come back to here, for instance, if you were to look at the specifics about my Scandinavian heritage, they say we cannot tell you whether it's Swedish, Norwegian, or Finnish. Now, I know that they're all from Norway, but they don't know that just from the DNA. So hopefully that answers that question. Now, we're going to do another little bit of interactive part here. So here is a match of some DNA. I want you to think through and predict what the relationship is between these two people which share this DNA. If you can't read, it's a little bit small. So if it's darker, which none of this is, it would be completely identical. If it's this lighter pink, it would be half identical. Gray is either not identical or not enough information. So I'm telling you, there's no completely identical sections in those. All right. So Julie guesses a parent-child, and that is correct. This is a parent-child relationship. This is actually my daughter and me. Okay. All right. Next one. How about this one? Now, hopefully you can see some of that dark, which is completely identical. The other that's half identical. All right, for the most part, people are guessing siblings. All right, somebody said fraternal twins. Fraternal twins would just be like siblings. So yes, it could be fraternal twins. If they're identical twins, it would be more, I mean, obviously there would be a closer match. But this actually is siblings. This happens to be my sister and I. All right. Now, how about this one? A little bit of a trick question. There's no DNA match in there. All right. This happens to be my wife. We're not kissing cousins. All right. So this is an arbitrary person, obviously my wife, who there's not the DNA relation there. If there had been, I would have been embarrassed to show it. No, I'm just kidding. But so this is how they're kind of doing it. And you can look at how close these things are and the different patterns. If you want to go really far back in time, you have to look at how frequently one it has been, or when did it first start showing up versus a more recent one. And it looks like, Martin, you've put a tool in here, which is, that's cool to know. I'll have to take a look at that. I'm not familiar with that one right off on my own. But that's how the Ancestry works. Again, comparing those different things. So a half sibling that's correct, Ahmed, there would only be 25% because they only have part of the mom, and then depending, or the mom or the dad, whichever one was the parent that's similar, and then those can be all kind of random. So it'd be roughly 25%, but you don't really know for sure what it is. And so there was a question as far as, going back and how, how does that work? Remember that the further back you go, the less precise it's going to be, because there's different events that can occur. The DNA can cross over, it can exchange things. There's different events that will happen within the cell, which is going to complicate that all. So that's when we're saying, well, it's roughly this similar, but it's not as easy to determine. All right. Let's look a little bit now at how these markers are used and how they can help us with medicine, or our pharmacology of the drugs that we would be taking. So again, if you study patients that have a particular disease, you can look at all of their different genetic markers, all of these polymorphisms, and you can see this, everybody that has this particular disease seems to have this particular polymorphism. And then you can, and statistically, you come up with the likelihood of that happening, and you start associating those markers with particular diseases. Now, these are just some that I found that I put on here of different diseases which are associated with different polymorphisms. I'm going to give you a couple examples. For instance, this was one you may have heard about, the BRCA. stands for the breast cancer gene. Now, there's a BRCA1 and a BRCA2. One is found on chromosome 17, 2 is found on chromosome 13. Now, there's lots and lots of different variants of these, but some of those have been shown to be associated with increased risk in breast and ovarian cancer. And they could be associated to some extent with prostate cancer as well. That's not as clear. So by looking at everybody, sequencing a bunch of people that have breast cancer, they can see this showing up and being a marker for the possibility of breast and ovarian cancer. So this happens to be one that popped up saying I had a more likelihood of ovarian cancer. Well, that's not going to be the problem. I could still have breast cancer. There are at-home tests where you can sequence or determine whether you have the BRCA genes. One thing I would caution you, though, you have to look, and I told you that there's over 1,000 variants in these genes. Some of these at-home tests only test for 3 of them. And so if you're only looking at a few, they may be the ones that are more likely, but that doesn't mean that you won't have it if you're negative for that. But nor does it mean that you will have it if you have one of these variants. And yes, Robert, men do have breast cancer associated with the BRCA genes as well. These variants that are associated with the breast cancer actually are found more frequently in the Ashkenazi Jewish heritage. Again, I just caution, we're talking about associations. Is it a given that you would have it? No, you don't know. You never know what exactly is going to happen. You can just say, I have a higher likelihood perhaps than somebody that didn't have that marker. Another case here is a gene called the ABCB1 gene, and it's actually associated with inflammatory bowel disease, such as Crohn's or ulcerative colitis. Now, there is a variant in the ABCB1 gene. It's located, I've highlighted it here on one of these sequences. And that one nucleotide change is associated with a greater likelihood of inflammatory bowel disease. So that's another example. And then the last set that I'm going to tell you about is markers associated with severe COVID infections. Now, ACE2, angiotensin 1 converting enzyme 2, happens to be the protein that the SARS-CoV-2 virus, which causes COVID-19, binds to. Therefore, if I have changes in that particular protein that make it better to bind to the virus, I'm going to have a more severe case of COVID-19. Or vice versa, if I have a change which makes it so it can't bind to COVID-19, that would do the opposite. I would have less frequency of COVID-19. They have found some variations that seem to be associated with expression levels of ACE2, but also some that have to do with the affinity of the virus to that receptor, which explains some of the susceptibility that people are seeing. So that's one particular case. There is also a gene called TMPRSS2, or transmembrane serine protease 2. This is also associated on the surface of these cells, primarily the ones that line our lungs and other epithelial tissues. So this gene or this protein is involved in actually clipping part of the protein on the SARS-CoV-2 virus, and that's required in order for that virus to enter into the cell. So again, if you have changes in that, that can change how much it gets into the cell or not, and they have observed some of that. Another one is this interferon-induced transmembrane protein 3. Interferon is associated with the immune system, and they have seen variations of it which are associated with increased severity of the disease. Fourth one I have here is called apolipoprotein E. And what they found is that 2 copies of ApoE epsilon 4 give you a higher risk of severe COVID-19. And then the last one here is I have the leucine zipper transcription factor, and there's a very minor allele that's not present in a lot, high percentage, but it has been associated with the respiratory failure that's seen in COVID patients. Now there's a question here, if you take an ACE inhibitor, does it reduce your chances of getting COVID-19? There has been some thought along that, but I have not seen any tests that actually, or clinical trials or anything like that, that really show that for sure. One thing you have to watch out for is where the molecule may be binding. If you take an ACE inhibitor and it's small enough, and I don't, there's too much unknown here, but if the ACE inhibitor is small enough, it may bind into ACE2, but still leave room for COVID to bind. So there's some different things there that we really don't know at this point. Anyway, I thought you might be interested in that. Again, there's a lot of people, this kind of shows you what people are doing, because as soon as you see some patients that have more severe disease and others that don't, they start looking at the DNA, start looking at some of these little teeny sequences to try to determine, can we find something that could help us understand why some people have more severe disease than others. Now, this last case that I want to show you with COVID-19 is actually a paper that just came out yesterday, and in fact is not it's not officially published until March 2nd. This is showing an association of Neanderthal DNA with COVID-19. And so what they were looking at is what sections of the DNA seem to be associated with severe COVID infections, hospitalizations. And they found this region on chromosome 12 in this OAS gene, the oligoadenylate synthetase gene, which happens to be associated with Neanderthal DNA. And what they've shown is that a single nucleotide variant that's inherited from Neanderthals seems to help protect against COVID-19. Now, I told you the Neanderthal DNA is often associated with how we interact with viruses, which is really interesting because we often think of, these viruses are just brand new. But so why would a Neanderthal DNA have any kind of association with how we would react with them? Now, this graph that I'm showing you here is the frequency of that single nucleotide variant from the Neanderthals in the world. Now, there's a project called the 1,000 Genomes Project, where they've looked at all kinds of different genetic databases and tried to look at these things. So each of these pie graphs is from one set of that genomic data. And what is dark red is the percentage that is this Neanderthal single nucleotide variant. So in these, like here, where you see a greater portion of that red pie, there's more of that Neanderthal DNA versus in Africa where there is none. Okay. Now, one of the things that really struck me as interesting here, now we have students both here in Nevada and students in Utah, so we've been looking at COVID cases in both states and all of that. I don't know if you've paid attention, but there is some discrepancy between the number of cases in Utah and the number of severe COVID cases if you associate that with deaths. Now, this set right here is from a Utah family database. So it's a whole bunch of DNA that's associated with that. Now, if you look at that, there's a high percentage of this Neanderthal DNA. In Utah, Utah ranks number 4 out of 50 states in the number of cases per capita. A lot of cases per capita. But they're number 46 out of 50 with deaths. It's kind of interesting. I'm conjecturing big time here. We have no idea why, but it is sort of intriguing that an area where there's a higher percentage of Neanderthal DNA is also a situation where there's a lower percentage of deaths per capita. So anyway, I just thought that was really kind of interesting. Again, this is brand new. It's going to be published in the Proceedings of the National Academy of Sciences on March 2nd, although you can read it online now if you're interested. So there was a question, Ahmed, you asked about correlation with the O-type blood. Yes, they have found that. I didn't include this in here because it's not really associated with polymorphisms, but yes, they have seen that. Okay, now to try to keep within the time frame, I'm going to keep moving forward here. Now we're going to talk about how those little changes in your DNA can impact how you respond to a particular drug. We call this area of study pharmacogenomics. Now, pharmacogenomics is looking at that, how genetic variations change our response to a particular drug. The goal of pharmacogenomics is to understand, what would be the best drug for a patient at the start of treatment. Now, ideally, you want to maximize effectiveness, minimize toxicity, avoid unnecessary treatments, but drug companies can also use it to help them optimize drug development. Now, if you look at the variability in drug response, there's a huge variability. If you look at this table over here on the right-hand side, I'm showing you the percent of patients where drugs aren't effective in helping them. So some patients it works and in others it's not. Now, there's a lot of different factors that are associated with this, environmental factors included, but it's estimated that about 75 to 85% of the variability in the half-life, that means how long the drug lasts in the body, is inherited. Now, let me give you an example. This is an example of a drug called antipyrine or phenazone. It was used in the US to treat pain and fever. It's no longer used in the US, although it is used elsewhere in the world. A study was done looking at twins and separating them out by identical twins versus fraternal twins. If you look at the average difference between identical twins in their response or their lowering of fever or pain, the average difference between those identical twins was 14 minutes. Whereas in the fraternal twins, which is on the right-hand side, the average difference was 300 minutes or 5 hours. So a huge difference there. And again, implying that that genetic difference is what really can impact how you would respond to antipyrine. Now, drugs interact with the body in several different ways. And the variance in the DNA can influence two different areas that we talk about a fair bit in pharmacy school, for instance. The variance can change how the body interacts with the drug. what the body does to the drug. So what we're talking about here, the big word is pharmacokinetics, which is saying how long does the drug last in the body? How fast does it get absorbed into the body? How fast does it distribute throughout the body? Is it metabolized more or less? And how fast does it get excreted? And that's determining how long that drug is going to last there. So the most common effects of these variants are changes in metabolism. Now, if you increase the efficiency of a metabolic enzyme, you're going to increase how much is removed from the body. If you were to decrease the efficiency of a metabolic enzyme, it's going to take it away. And I'll show you that in a little bit. The other way that variants can influence how a drug would interact with us is in an area called pharmacodynamics. Now, pharmacodynamics is talking about how the drug can interact with the body. And specifically, we're looking at things like the target of the drug. So if we were to change the target of the drug, we already talked about one of those. So if we changed ACE2, that could influence how COVID binds to a cell. So that would be the target of the drug. We can also change how our immune system interacts with it. That would be similar to the interferon one that we talked about with COVID-19 as well. Now, the FDA is actively looking at this, and they estimate that roughly I most recently heard 10 to 15% of drugs will actually have some kind of label associated with them that will impact the pharmacogenomics, or has information of pharmacogenomics. I said that kind of funny, but the information that we have from the DNA would impact how we would dose or how we would treat with that particular drug. Now, there's two places where you can go to find information about these. One of them is called CPIC, or the Clinical Pharmacogenetics Implementation Consortium. This is a group of volunteers that are actively looking through all of the literature and trying to say, okay, what things do we actually have that will create an actionable way that we should treat differently based on the pharmacogenomics. So they have these sets of guidelines, and they say, okay, if you have this marker, that tells you this with this particular drug. Now, the FDA also has a list of their pharmacogenomic associations. Interestingly enough, these are not exactly similar. The FDA one is more extensive, likely because they include things that may impact therapy, whereas the CPIC is saying this is something where we know that it is a critical thing and this should impact, you should do something, versus the FDA saying, well, this looks reasonable. So if we look at the CPIC guidelines, currently they have narrowed it down with what's on their list, 20 different genes that I've listed here that impact 65 different drugs. All right. So there's some that interact with the immune system. I'm going to talk about those a little bit more. There's some others. These are more like the targets of some of the different drugs. The cytochrome P450s are involved with metabolism. I'll give you a couple examples of those. And lastly, we have a number of other metabolic enzymes that do some different things. One of them we're going to talk about is one that has to do with statins. But I'm not going to do all of these examples, or we just simply do not have time for that. But if you're interested, you can go to the CPIC websites and you could read their guidelines. They have information there, including information about how frequent some of these things are found in different ethnicities and that kind of thing. Now, the reality is we're looking at a small number of drugs and where just one gene is impacting it. So from this paper, if you look at it, in these numbers that they used, they had 1,200 different FDA-approved medications, and they found that 10% of them had actionable pharmacogenetic information that they could be used. The other 90% didn't. Now, if you look at prescriptions, it's a little bit different here, because in 4 billion prescriptions, 20% of those do have something that could be influenced by the pharmacogenetic or pharmacogenomic information. But there's still 80% of them that aren't going to be impacted by that. However, and I was just went to this virtual summit last week. And one of the things that they pointed out was this study where they were looking at a whole bunch of patients that they screened in a hospital system. And from their studies, and this actually, there were multiple people that have done this and they come up with the same numbers. They showed that 99% of screened patients have the high-risk genotype in at least one of 12 different CPIC genes. So that means they have 2 copies of a variant which could be impacted by drugs. Now, the discrepancy here is they may have one, but whether are they prescribed that drug or not? They may or may not be ever exposed to whatever drug is influenced by that. However, the frequency of prescribing drugs which are impacted by pharmacogenomics increases with the age of the patient. So they're exposed to a lot more pharmaceuticals and a lot more of the high-risk pharmaceuticals. So it's something to be aware of. Regardless of what percentage of it is there, if you're in the 10 or the 20% of these pie charts that I have here, you would be interested in knowing whether your genetics is going to impact it. So let's talk about a few examples here. Before we do that, I want to talk just a little bit. We're going to talk about some of the metabolism ones. And I just want to show you briefly how the metabolism works. I mentioned this earlier, but I want to do it a little bit more specifically. So if we take an active drug and we have a metabolic enzyme, that metabolic enzyme could turn it into an inactive drug, so it gets excreted or something like that. Now, when we talk about metabolism, we really talk about 4 different groups of people. We have what we call ultra rapid metabolizers, normal or extensive metabolizers, intermediate, and poor metabolizers. And I've shown you over here what can happen with some of the different genes and how that can give you an ultra rapid, extensive metabolism, that kind of thing. But it's important to know that extensive or normal is what generally, that's what they're looking for when they do clinical trials. They're saying, okay, we're going to dose for a normal extensive metabolizer. Some people are going to be ultra rapid. They're going to metabolize even more. Others are going to be poor metabolizers and will go even less. So in the case of this situation where an active drug is converting it in, I said that really funny. The metabolic enzyme is converting the active drug into an inactive drug. If you are an ultra rapid metabolizer, you need a higher dose because you're not going to have as much drug in your body as a normal metabolizer because you're metabolizing more. Whereas if you look at a poor metabolizer, they're going to have too much drug in their blood. And there's a greater possibility of side effects happening in that situation. Now, it gets a little complicated because some drugs are prodrugs. Actually, for sake of time, I'm going to skip that right now. But I do want to show you this. This is different drugs which are metabolized by cytochrome P450s, which are the main metabolic enzymes. Now, the ones that I've highlighted in red here are in the top 200 drugs. So you see there's a lot of very frequently prescribed drugs which are impacted by the cytochrome P450s. Now, let me show you cytochrome. We'll talk just a little bit about one of them called cytochrome 2D6. Now, there's different alleles of CYP2D6, some of which have a loss of function. So star 3, star 4, star 6, 7, and 8, don't worry about how those are named at this point. Those all have less function of cytochrome P450 2D6, CYP2D6. There's others that have no function, it doesn't work at all. And there's other ones where they have extra copies of CYP2D6, and so there's more rapid metabolism. Now, if you look at the population frequencies, Middle Eastern and Oceanian are more likely to have ultra rapid metabolism, meaning they have extra copies of CYP2D6. Whereas Caucasian population is more likely to have poor metabolism, although it's 6% and it's subtle differences in that group of poor. Now, there's two drugs that we're going to talk about here, codeine and atomoxetine, that I'll show you in relation to CYP2D6. All right, so codeine and CYP2D6. Codeine is metabolized by CYP2D6, and it removes this methyl group, which turns it into morphine. And that's how codeine is gives a pain relief, is through the morphine part of it. Now, if you have extra copies of CYP2D6, if you're an ultra rapid metabolizer, you have the potential of a morphine overdose because you convert more of the codeine into morphine than a normal person. Where we really see this playing a role is in nursing infants, because the morphine can be passed from the mother to the child. And there actually have been cases of death in infants from morphine overdose purely because the mother was taking codeine. So what does that mean? Well, if you're an ultra rapid metabolizer, you should avoid codeine because you have the chance of converting more of it into morphine. Or if you're a poor metabolizer, codeine's not going to really do much for you because you're not going to convert it into morphine. So that's one example. Atomoxetine is similar. It's used for ADHD. And again, if you are a poor metabolizer, you're going to have a higher concentration of atomoxetine in your blood. So ultra rapid metabolizers aren't going to get any benefit from it. If you're a poor metabolizer, you're going to have a lot more side effects, which include sleep disturbances, depression, tremor, and such, as I've listed there. Now, let me give you a personal example here. So I've introduced you to my wife and daughters and all that, and a grandchild and pictures. This is my mother and my father. Now, a little over 4 years ago, my mother went in for a back surgery. It was expected that she would recover fairly quickly from that, everything would be fine. But when she came out of the operating room and they gave her pain medications, my mother was experiencing some very weird symptoms. And those of us that were around her were observing these and kind of wondering what on earth was going on. The scientist in me was even wondering more. So she was seeing things, hallucinations. She was really confused. She would be working with things with her hands and think that she was doing other things. It was like she was knitting or things like that with her hands, and she thought she was really doing it. And it was really kind of confusing. And so I teach at a pharmacy school and I'm a scientist. I'm like, well, what drugs is she taking? Well, this is some of them. I'm not going to show you all of them, but there were 3. She was on tramadol, which is an opioid for pain, which is a very common thing to be given after surgery. She was on diltiazem and ondansetron as well for nausea and blood pressure. Okay. Now, I was like, well, side effects of these: blurred vision, dizziness, nausea, hallucinations, and such. Well, are these associated with what's going on? And I was scratching my head trying to figure out what was going on. And I happened to be talking to Dr. Talbot, who teaches here at Roseman as well. And I'm like, I don't know what's going on. And he goes, well, you know that all but one of the opioids are metabolized by CYP2D6. I was like, no, I guess I didn't know that. And so I started taking a look at it, and lo and behold, diltiazem, tramadol, and ondansetron are all metabolized by cytochrome P450 2D6. So in this situation, if my mother, the patient, was a poor metabolizer at CYP2D6, she would have a higher concentration of the drug than would be expected. Therefore, she would have higher side effects. And remember, I showed you previously that star 3, star 4, 6, 7, and 8 are all loss of function, so they would not metabolize, you would have the higher concentration. Well, this is actually the first thing that led me to try to do one of these direct-to-consumer genetic tests. So I sent my spit off to 23andMe, and I got the results back, and sure enough, I have a copy of star 4 CYP2D6. Star 4 has no function, no activity in it. So I inherited one copy of 2D6 that is no good, and I'm pretty certain I know who I inherited it from. I think that was from my mom. So anyway, that's just an example of how these things can help you out. Let's do another one, and then we're going to wrap up here pretty quick. CYP2C19. This is associated with a drug, a very common drug called clopidogrel. There are different alleles of it which have a loss of function as well. Those poor metabolizers are primarily found in East Asian and Oceanian, East Asian or Asian and Oceanian populations. So the issue here is that in order for clopidogrel to actually work, you have to have it be metabolized. And if you're a poor metabolizer, you're never going to get any effect from clopidogrel. And so the FDA has put on in the labeling for this drug that if you're a poor metabolizer, you're going to have a higher incidence of heart failure because you're not getting any effect from the clopidogrel. Now, why is that important? Well, we have other drugs that can be used which are not affected by this. And so if you knew that you were CYP2C19 poor metabolizer, you would not want to be put on clopidogrel. And one of the speakers that I heard last week was talking about how she had to fight, I shouldn't say fight, she had to try to explain everything to a physician who wanted to put her father on clopidogrel, but he was a poor metabolizer at CYP2C19. So she was pushing to try to get a different drug. For sake of time, we went a little bit long. I'll briefly talk about this one. I'll have the slides up here. It will be in the video recording. You can look at the slides more. OP1B is a protein which is made by this gene called SLC01B1. It is involved in removing drugs from the blood into the bile. We have poor activity, which if it's poor activity, it's not removing as much drug into the bile, so there's higher concentration in the bloodstream. We see that in these populations here, so Caucasian, East Asian, Middle Eastern, and Hispanic populations. Why is this important? Well, all of the statins, with the exception of fluvastatin, are actually removed from the blood by OP1B1. Now, it really plays a role in simvastatin or Zocor because simvastatin requires a really high dose, and the threshold, the difference between a toxic dose and an effective dose is not very big. All of the statins, if you had too much of them, can cause myopathy, which is a decrease in muscle tissue. In particular, so if you look at a person that has 2 bad copies of OP1B1, the concentration in the blood of simvastatin can go up over 220%. So it's a huge increase in the blood and much more likely to cause myopathy. There's another one here, which is HLA-B. HLA-B is involved in presenting things to our immune system, so it triggers an immune response. Carbamazepine is a very common drug. It is influenced by HLA-B. The problem with this is carbamazepine binds to HLA-B and causes this immune response. And that immune response can cause these rashes. It's called Steven-Johnson, or the 10. It's very severe and can be fatal. I can't imagine having this happen. Another drug which can interact with HLA-B and do the same kind of thing is allopurinol, which is, again, another drug that's used commonly for gout. Why clinically is that important? Well, we have an alternative. We can use febuxostat, and it doesn't have the same kind of effects. So if we know that we have the possibility of an issue, such as in Asian population, it would be better to treat with febuxostat instead of allopurinol. All right, I'm not going to go through how you can do the raw data. Just in summary, let me just say that we've talked about how those changes in that DNA, if you look at groups of them and how those are related to other things, we can identify, we can get similarities from one person to another, or from the effect in a drug to something else, and that can allow us to understand better how we can go about treating differently with drugs, or the relationships with those people around us. So most of the drugs, most of the pharmacogenomics data doesn't lead to a risk of life, and so we don't consider that an actionable pharmacogenomics, because it's not, I mean, if I'm an ultra rapid metabolizer, and what that means is I have a lower concentration of the drug, I just will need a higher dose of it. If it's not life-threatening, that's not a big deal. But if I have too much of the drug and it's going to cause a lot of issues, that's when it's a big deal. With that, I'm going to end, and I'll glance. Vanessa, have you had any questions in particular?
No, there are lots of comments in the chat. Maybe while you're looking at those final questions, I'd just like to remind everybody that our next event is on March 18th, when Dr. Farbman, one of our neurologists from Roseman Medical Group, will be talking about dementia and cognitive decline, how we properly diagnose and treat these diseases. And he will be paired up with Michael Kling, who is an estate law and planning attorney, who will be talking about how we protect our loved ones when they are diagnosed with dementia. So with that, I'll turn it back over to you, David.
Okay, there's a couple questions here. Julie, you asked about how is this routine or not? Part of the discussion that this seminar series that I've been going to has been talking about the push to try to have a more general pharmacogenomics panel done to try to see how people are, what things they're carrying and what drugs should be used. And so there's a group, the North Shore in Chicago, for instance, where all of their patients that they come in, they try to do the genetic testing on them and keep it as part of their electronic health record. There's not widespread acceptance of that, not widespread implementation of that yet at this point. You said it seems like it would be good for everyone. I think that there's value in it. We do have to be careful on where it's done. I mean, I am a consumer with 23andMe data. Is 23andMe the best one for pharmacogenomic testing? Probably not. There's other ways of doing that. You may need to do several of them to try to see, because not every test is 100% accurate. So there's some caveats with it. There's a lot of discussion that we've been having in the seminar as well on the reimbursement for it. Will insurance pay for it? And that kind of thing. And so that's a little bit more tricky. So I'm not sure if it's Linus, or I'm not positive how to say the first name. I may have said it wrong there. Question about DNA tests being used to design the ideal child. That I think is still pretty far-fetched. I mean, we already do, there's the possibility of amniocentesis where we look to try to see if there's anything perhaps wrong, if we know that there's a chance of it in a fetus. But actually designing the ideal child, that's still a long way away before we would do that or be able to do that. And you also ask about baseline DNA tests becoming a regular part of medical histories. There's a lot of people that are saying we should do that. At least with the key genes that we know of that are impacted, or that drugs are impacted by, we could or should perhaps have that in our electronic records. There's a lot of other people that would be concerned about all of the other genetic information. So it would probably be more comfortable for them if we kept it to a smaller set. All right. I don't know if there are any other questions. If you had any, feel free to unmute.
David, I think I speak for everyone in thanking you for an incredibly fascinating discussion. We so appreciate you, and you look exactly like your dad. So that was
That's the genetics, right? I fit in my genes. All right. It's wonderful to have been with you. I hope that it's been informative. Like I said, there's all kinds of things that we could have talked about. It was hard to try to narrow it down, but hopefully it's been good for you. Thanks for joining us.
Thank you, and good night, everyone.